A month ago, I received a request to make a video on DNA extraction. Now, the time has come. DNA, deoxyribonucleic acid. DNA is a polymer composed of two polynucleotide chains that coil around each other to form a double helix. DNA was first discovered in 1869 by Friedrich Mischer. DNA is found in every living organism. It's responsible for carrying and transmitting the hereditary materials of the genetic instructions from parents to offsprings. I know you're familiar with the model of DNA, but have you ever seen an actual DNA? No? Well, you'll see today. So, including this, there will be 2 videos on my YouTube channel on extraction of DNA. In the first part, or in today's video, I'm gonna extract DNA from banana. And in the second part, I will extract DNA from my saliva. However, today I'm gonna extract DNA from banana. To do so, I'm using the following materials. A conical flask with 100 ml of water and a beaker also with 100 ml of water. 87% ethyl alcohol or ethanol, 5.85 grams of sodium chloride or table salt, an empty beaker with a tea filter, liquid dish soap and food color, 2 pasteur pipettes, 2 test tubes, a glass rod, test tube holder, a blender machine. The final and the main ingredient is a banana. At first, I have taken around 20 ml of 87% ethanol in a pot. To extract DNA, alcohol must be ice cold. So sealing the pot well, I put that in refrigerator for 2 to 3 hours. In the meantime, I've prepared 1 mole solution of sodium chloride or table salt by dissolving 5.85 grams of table salt in 100 ml of water in a conical flask. The salt solution is ready now. Then I peeled the banana and sliced that. Then I dumped the sliced parts in the jug to blend. Here I go. Then I'll pour all the banana juice into the beaker through a tea filter. Now I'm adding the salt solution and a little bit of liquid dish soap into the banana juice, stirring gently to mix all the stuffs. Then here I'm taking around 5 ml of the mixture in a test tube. In another test tube, I'm taking around 6 ml of 87% ice cold ethanol. Then I added some yellow food color drops into ethanol. Now all I have to do is just to add the ethanol into that mixture. As I added ethanol, there has been formed 2 layers in the test tube. The bottom one is the layer of the mixture which contains banana juice, salt water and liquid dish soap, and the upper layer is of ethanol. As you can see, there are lots of solid stuffs floating around in the ethanol layer, and those solid stuffs are the extracted DNA of banana. The chemistry behind this is very important to understand. Generally, DNA is insoluble in any alcohol, but in ice-cold alcohol, DNA is way more insoluble, and that assists to extract DNA from human or plant cells perfectly. Anyways, to extract DNA from banana, first I added salt water, then I added liquid dish soap, which breaks down cell membranes and release DNA. The salt helps bring the DNA together, and the cold ethyl alcohol or ethanol helps the DNA precipitate and come out of solution. I added yellow food color in ethanol so that viewers can notice the different layers and the DNA perfectly. Instead of ethanol, rubbing alcohol or isopropyl alcohol can be used to extract DNA. But I don't have that one in my lab. However, this is how you can extract DNA from any fruits or plants. In my next video, I will extract DNA from my saliva. Till then, stay tuned with your Chem Mate.